Welcome to Genetics for Probability of Different Genotypes. We will be utilizing the Punnett Square and also the branching method as well. For illustration purposes, we're going to be utilizing the polled versus horned gene. The polled gene is dominant over the horned gene. So if you're homozygous polled or you've got one copy of the polled gene and one copy of the horned gene, the animals are going to be polled. For our illustration, we're mating two individuals. We're assuming they're both heterozygous for the polled and horned genes. It is common practice to put the males on the top and the females on the side. In this case, it really doesn't matter in that both are alike genetically. They're heterozygous for polled versus horned. So first, we're going to let the genes from the bull flow down into our Punnett Square. So we've got the polled gene on the left here. Now the horned gene, it's lowercase. It's a good idea to always put the lowercase to the right in your squares and put the uppercase to the left. And then we just follow through with the genes from the female. And we've got the Punnett Square filled out. And as you can see, 3/4 of the offspring would be expected to be polled. Of those, 2/3 are going to be carriers for the horned gene. And you got a 1/4 chance of the individual being horned, containing two lowercase horned genes. In our example here, we're going to have a homozygous polled bull mated to homozygous recessive little p or horned cow. So homozygous polled bull, two copies of the polled gene mated to horned cows. And what are we going to get? As you can see here, the bull is going to give the polled gene to each of his offspring. The cow is going to give the horned gene to each of her offspring. And because we're getting the polled gene from the bull, the horned gene from the cow, all the offspring are going to be identical. Would expect all of them to be polled, but all of them to be carriers for the horned gene. We have a similar example here, but we're using the black hair color versus red hair color. Black allele is dominant over the red allele. So we just fill in our boxes again for the Punnett Square, and we find that would expect 3/4 of the offspring to be black. Both parents were black, but carriers for the red gene, and you get a 1/4 chance of the offspring being red. Now we're carrying the concept just a little further. Previously, we're just mating two individuals. In this case, we may be mating a bull that's heterozygous for our traits here. He's got the big P and little p, and he's got a 50% chance of donating the polled gene, 50% chance of donating the horned gene. And he's being mated to a group of cows. And we know in this group of cows, probably because of their pedigrees, that we expect within this group of cows, most of them are horned. We've got a few polled animals. In fact, our calculations are that within this group of cows, 20% of the genes are going to be polled, 80% are going to be horned. So the first thing we do here is just fill out the boxes with the horned versus the polled genes. Because the frequency of the genes in our cow herd was not 50-50, but instead 20% and 80%, we have to multiply these frequencies through. So 0.5 times 0.2, you've got a 0.1 chance or 10% chance of having a homozygous polled individual born. And for this square here, it's going to be the 0.2 times the 0.5, 0.1 here. This square down here is the 0.5 versus 0.8, 0.4. And here we have a 0.8 times 0.4. So in this particular case, we got a 40% chance of getting individuals that are horned. So 60% of the chance, 60% of the calves would be expected to be polled, and 40% would be expected to be horned. So what we have here is what's called a double Punnett square. We've got two traits now. They're two independent traits. We've got a male that is heterozygous for both the black versus red coat color and polled versus horned. And we're mating that individual to a female that is heterozygous for both traits as well. If you remember previously in this chapter, we said that if an individual has two traits, heterozygous for both, we have four possibilities as far as in the gametes. So we have the sperm on top here, and we have the possibilities in the eggs on the side. And then we're going to have to go through the work of filling out the Punnett square. You have to be very careful here. We need to get the gametes correct, and then we need to fill out correctly in our squares. And you've got about 80 times here, 80 chances of making a mistake. So you do need to be very careful. So first we'll start out by letting the male genes flow down through the squares. So in this slide here, we've got all the male genes flowed down, and good idea, keep the uppercase to the left, keep the lowercase to the right. And in this case, we have the colored genes on the left, polled versus horned to the right. Really important to try to keep things really organized so it's easier to count the genotypes when you get finished. So here we have all the squares filled in from both the male and the female side. And then we can start counting. So homozygous dominant at both sites, well, we just have one. And maybe we can just go down through our squares. We've got two that are homozygous for black, polled but carriers for the recessive horned genes. So that one's right here. And then we have one that's homozygous for black and homozygous for horned. That's this one here. We've got two of these, heterozygous for coat color, homozygous polled. That's these two. Down the diagonal, we've got individuals here that are similar to the parents, heterozygous for both traits. So that's these four. So the most common one is what the genotype of the parents, and we can continue on, and hopefully these add up to 16 squares. But as you can see, it's pretty complicated, easy to make mistakes, so you have to be relatively careful. Rather than doing a double Punnett square, if you've got two traits, I think it's always easier just to think of them as two independent traits. Do a Punnett square for each one, as we have here, and then put this over onto a branching method. We know that from single Punnett square that 1/4 of the individuals are going to be homozygous, the big E, 1/2 are going to be heterozygous for that coat color, 1/4 are going to be two little e's. And we've got the same frequencies at polled versus horned. So for those individuals that are homozygous black, 1/4 of those are going to be homozygous polled, 1/2 are going to be heterozygous, 1/4 are going to be homozygous horned. Of the individuals that are heterozygous for coat color, we know that 1/4 are going to be homozygous polled, etc. And we can just fill in our chart here. So what percent are going to be homozygous dominant at both rates? It's just 1/4 times 1/4 is 1/16. What percent are going to be red and horned? It's just 1/4 times 1/4, 1/16. And then we can check ourselves, need to add, if we add all these fractions together, it has to equal 1 or 100%, and in fact it does. A lot quicker, a lot easier to get all those geno frequencies this way rather than doing a double Punnett square. Rather than having interest in genotypic frequencies, you might be more interested, and many times we are interested in, what are the expected phenotypic frequencies. So we know that from our Punnett square that 3/4 are going to be black, 1/4 are going to be red. Of the black ones, 3/4 expected to be polled. So what's the chance of being black and polled? It's going to be 3/4 times 3/4, 9/16. Chance of being black and horned, it's just the 3/4 times the 1/4, and that equals 3/16. And we just continue to fill it out. So we've got 4 possible phenotypes. We have the probability of getting each of those 4 phenotypes. Branching method here is just a lot easier again than doing the double Punnett square. Well, that concludes the little video on using the Punnett square and branching method. I hope this video has been found to be beneficial in understanding gene and phenotypic expected frequencies.